Good morning, everyone. I am going to explain how proteins are created, the process of protein synthesis. Before we start off, there's two main things we need to know. First off, proteins are made up of long chains of amino acids. That's how they are made. Secondly, we need to understand base pairing. In DNA and RNA, there are four types of base pairs. In DNA, there is cytosine, thymine, adenine, and guanine. For the purposes to make it short, we're just going to call them T, A, G, and C. In mRNA, they have what's called uracil instead of thymine, but it acts the same. It still bonds with adenine, or the A. So first off, protein synthesis has two basic steps. First is transcription, second is translation. For transcription, first off, the cell realizes that some proteins need to be made for some reason or another. The DNA understands this and will unravel or unzip so that the mRNA can come in and create a complementary strand. How this works is, for example, let's say the DNA unravels and the base pairs, there are adenine, thymine, and cytosine, or ATC. The mRNA comes in, and because of the rule of base pairing, it will have a T, an A, and a G, which are the exact opposites of A, T, and C. So once this happens, the mRNA comes in and makes as long of a strand as it needs to in order for the synthesis to occur. After that, the mRNA goes away from the DNA, and the DNA seals up, good as new again. The mRNA will exit the nucleus and go out into the cytoplasm, which is pretty much the housing inside of the cell for all of the organelles. It's kind of squishy, just, instead of air, it's that thing they float around in. Anyway, the second step is translation. Once the mRNA is outside in the cytoplasm, it will go to one of the organelles called a ribosome. The ribosome is, you can pretty much view it as a factory for where proteins are made, all right? The first step is the mRNA will slide into the ribosome, and then tRNA will come. The tRNA is a set of three base pairs called codons connected to an amino acid. Now what happens here is different sets, triads, these codons, they each code for a different amino acid. There's lots of charts that can show you how this is done. So what will happen, the mRNA inside of the ribosome will pair up with the codons in the tRNA. That will happen, so let's say it's sitting there, tRNA comes for 3, it'll slide along in the ribosome, another 3 will come, another 3 will come, and as that happens, the tRNA will release the amino acids so that they will be stuck together in the specific order that the mRNA codes for. Once that happens, the protein is as long as it needs to be. A stop codon will come along. That is a codon where instead of coding for a specific amino acid, it will tell the ribosome to stop and release the protein because it is now finished. And that's what happens. That's how proteins are made. This can happen as many times as needs be because the DNA is not damaged in any way by what happens with the mRNA. It can unzip and rezip as many times as it needs to. So, really interesting process there. Hope you enjoyed.